Hi, good afternoon, everyone. Thank you for being here. First, let me thank Soft Matter Editorial Board for this incredible honor. The journal Soft Matter has been a big part of my career, starting from my grad school and to my independent career. So it's really special to be honored by the same journal. And I'm really excited to be talking about our work on diffusive forest enhanced instabilities. And before I go into the details, I would first like to thank my group and funding sources that have enabled this work. And the work that I will be talking about today was primarily led by Siamak and was supported by Lucas, who did some experiments on our work. All right, so I like to do this in all the talks that I give is that if you don't remember anything else from my talk, I want you to remember three things. And those are these. Uh, what is diffusivephoresis? So if you're not familiar with this phenomena, what exactly is diffusivephoresis? And then I'll talk about two different instabilities that diffusivephoresis can enhance. One of them is a Rayleigh-Taylor instability, which is going to be my second topic. And then the third is that diffusivephoresis can play a role in patterns on a fish, which are basically reaction diffusion instabilities. The articles that sort of go into the details of these work are listed below. One of them is still under preparation, but the other two are out if you're interested to learn more about the details. All right. So with that in mind, I'll start with the first topic about what is diffusivephoresis. So a quick show of hands, how many of you have heard this term before? All right, a few of you, but not all. So let me try to kind of break it down. Diffusive phoresis is essentially diffusion plus phoresis. So it's made up of two words, diffusion plus phoresis. So it's a response of colloids to concentration gradient of a solute. So imagine there is some background diffusion of a solute, and then there are colloids dispersed in it, and these colloids are essentially responding to this diffusion of a solute in the background. So it's the movement of these particles because of diffusion of something else. And these particles can be anything. So these can be, synthetic particles like polystyrene. They can be DNA molecules like DNA origami. You can also think about biological swimmers. And then in solutes, you can have electrolytes like NaCl, bases, NaOH, acids, HCl, and so on. And you can also have non-electrolytes like sucrose, which can also kind of create this sort of solute. So there are many different kinds of particles in solutes. So now let's try to make it more concrete. Let's imagine that we have a microfluidic channel where you have some flow going in the main channel, and there is a dead end pore that is coming out of the main channel. And now because it's a dead end, you cannot have any bulk flow going through this pore. And imagine that you have been able to sort of, create this scenario where we have high sodium chloride concentration in the dead end pore and low sodium chloride concentration in the main flow. Now, because this is a dead end, as time goes on, essentially sodium chloride will diffuse outwards. And again, there is no bulk convection. So it will primarily be diffusing outwards. And now because this is diffusing outwards, let's imagine these are polystyrene particles. They can actually go into the pore by this phenomena of diffusivephoresis. So again, it's a diffusion of this salt and the particles are moving in response to it. Now, this exact phenomena you can actually do it in the lab. And so in this video that I'm about to play, what you will see is the fluorescent polystyrene particles, but you will not see the sodium chloride concentration. So just imagine it's going sort of out of the pore. So it's sort of higher at the end of the pore and then lower at the start of the pore. So let's play a quick video. Uh, and you can see these particles are going into the pore. And as you can see, the theory is actually able to do a really good job in capturing what's going on in this phenomena. I wanted to note that the particles are moving almost like together, like they're moving in a band. I'll come back to the signature in a minute. So the first question that I always get asked a lot is why does this diffusivephoresis occur? It's a complicated question, but let me try to kind of simplify this phenomena. And primarily, if you think about diffusivephoresis, it has two mechanisms. One is electrophoresis and one is chemiphoresis. And the combination of the two gives rise to diffusivephoresis. Electrophoresis is only present with electrolytes, but chemiphoresis is present always. And so now let's try to imagine this conceptually. So imagine now you have salt. Again, you can think of NaCl if it makes easier. And let's imagine we have a positively charged particles. So the positive signs are given by blue, and the negative is, the green sign. And let's again imagine that you have a concentration gradient with high being the left side. low being the right side. Now, so what is the electrophoresis part is that if my ions have different diffusivities, so let's imagine that the blue ion has a higher diffusivity, so it'll try to move faster. And now it's a positive ion. So I need to kind of keep the positive and negative ions together. So imagine if you have sodium chloride, I cannot have sodium ions outrunning the chloride ions. I need to keep them together to maintain electron neutrality. As an electric field develops in the opposing direction of concentration gradient, and then this electric field can actually electrophoretically move the charged particle. So that's the electrophoresis. In contrast, if my green ion is faster, then it is going to basically kind of has to be kind of slowed down. And because it's a negative ion, electric field will develop in the direction of concentration gradient. So they can slow the negative ion down and keep it together with the blue ion. And so this is the phenomenon electrophoresis. Naturally, this is only present when we have electrolytes. The other phenomena is chemiphoresis. In chemiphoresis, essentially you attract an ion near the surface or repel the ion near the surface. So now this is the case of attraction. So what you're seeing here essentially is basically you have these ions being attracted to the surface. So you have much more ions present near the surface. In the case of electrostatic interactions, these are going to be the oppositely charged ions. And so essentially what you see here is that basically we have high concentration of green ions in the surface, moreover high concentration near the surface and to the left than the right hand side. So we have higher concentration to the left compared to the right, and so naturally you will have higher pressure because of osmotic pressure. You have higher pressure on the left than the right. Now imagine you are in the particle frame of reference. In that case, the flow will happen from left to right, right? Because you are in the particle frame of reference. So flow in the fluid will essentially happen from left to right. But actually, the particle is moving and the fluid is stationary. So the particle will move from right to left. And so that's why the flow will happen towards higher concentration. So this is known as chemiphoresis. And so basically, we get this phenomena where diffusive phoresis is the combination of electrophoresis and chemiphoresis. Now, in non-electrolytes, essentially, you have a different kind of interaction, which can again increase the concentration of ion near the surface or decrease the concentration of ion near the surface. And that will dictate the sign of diffusive phoretic velocity. All right. So now diffusive phoresis has a signature, and this is basically what I like to call as banding. So I want to give you this example, which is a very beautiful experiment that came out almost 10 years ago from Todd Squires group at UC Santa Barbara, where they took this hydrogel, created this beautiful 1D gradient using HCl, NaOH, and NaCl. So the idea was to create a pH gradient and a salt gradient. And in some conditions, what they were able to see was the particles basically were initially uniform, and they all kind of sort of moved toward the middle and created this band in the center, a beautiful focusing region in the center where they'll basically stay there. We were actually able to reproduce this qualitatively in our numerical calculations. And what we found was that the pH dependence of the zeta potential or the charge of these particles was very crucial in kind of obtaining its phenomena. There have been other also work from the Squires group, which is this is beautiful experiments using hydrogel post. So these are 2D experiments. And what you will see again here is you can create this beautiful band of particles in which dissolves over time. And sort of this is something that they report in their experiments. We were actually able to report something similar in our this sort of computational model where we were able to create the band and then it dissolves over time. And this is the phenomenon we call as pseudo-focusing. Now, this signature where the particles tend to create these sharp gradients within a diffuse system is the signature of diffusive phoresis. And the reason we call it negative diffusion is that that's typically something you will not expect in a diffusive system to have sharp gradients. So what's happening is these colloids are moving at the speed of diffusion of the solute, but they're much bigger. They're about a micrometer in size, give or take. And so they don't have their diffusivity of their own. So they kind of stick together as themselves. So this is a very clear phenomenon and a signature of diffusive phoresis. So what can we do with this signature? So with that, now I'll go to the first part of the story is that we can use the signature of banding to trigger an instability. And this paper was basically inspired by this idea of Rayleigh-Taylor instability. So Rayleigh-Taylor instability, if you're not familiar with it, just a very simple idea is that if I have a heavier fluid on top of a lighter fluid given by density, and if gravity is acting downwards, if I give some perturbation to the interface, basically these perturbations can grow with time and I can create this instability. Now you can look around for YouTube videos. So for instance, you can look at this YouTube video. This is not a experiment basically that you can, this is not a controlled experiment, but it will give you an idea. You've just turned around. So the clear part here is the canola oil. The blue part here is the water. And there's some surfactant mix to kind of, control the interaction between the two. And you can say you can create this instability known as Rayleigh-Taylor instability. You can also numerically kind of create this instability in your codes. And you can again see these beautiful instabilities arising. So this is happening because of density difference. So what does this have to do with diffusivophoresis? So what we did was we thought about an idea inspired from a prior work in this area is that we will use a membrane to create diffusivophoresis and then use a diffusivophoresis to trigger this instability. So what's the idea? So think of a very simple thin channel, a Healy-Shaw channel. Just imagine we filled it with water and sodium hydroxide or sodium chloride, and we suspended polystyrene particles uniformly through it. Now, if you put the membrane on top, what happens is, in this case, if we put a proton exchange membrane, is at the top, let's imagine you had NaCl. So at the top, it will become HCl because all the sodium ions will get exchanged by the HCl ions, H ions. So it becomes HCl. So HCl at the top, NaCl at the bottom, and again, you have a concentration gradient of an electrolyte. Now the polystyrene particles are charged. So you will again move via diffusivophoresis. Now we have done controlled experiments. You will not basically see any kind of major movement if it was just pure sedimentation. So this is actually diffusivophoresis and we can control it. So now again they will move, they'll create a band, and because polystyrene is denser than water, essentially that band will have higher density than water that can trigger an instability. So this is what you'll see. And you can see this beautiful sort of comparison between experiments in the model that we are able to see. This is with electrolyte NaOH. So now first point, the simulations were done using multiphysics simulation, where we are solving for concentration of all the ions. Then we are solving the particle concentration with diffusivophoresis velocity including in it, while background fluid flow is also being solved. Now this is like a Rayleigh-Taylor instability. Basically, this is moving with a front as well. So it's slightly different. But again, we are using a diffusivophoresis as a trigger mechanism to create this instability. And again, if you do a control experiments, you will not see the similar phenomenon. Next, so what can we do with this data? So first thing is to track that open area at the top where there was no particle left. So we call it the exclusion zone. And so this is dEz. So this is dEz square versus time. And essentially, you basically see a linear line, which basically shows that it's a diffusive front. Basically, it's going like a diffusive front. And then we have two salts. So NaOH, as you can see, is 4 times faster almost than NaCl. And so why is that happening? So there are a couple of reasons. But I'll try to simplify that to give you an example. So let's imagine we have NaOH at the top. If you have NaOH, then again, near the membrane, all the positive ions will become protons. So this becomes water at the top. It's H plus and OH minus. It becomes water at the top. So if you think about the gradients, I have Na plus at the highest at the bottom, OH is the highest at the bottom. So both the ions are highest at the bottom. And when I think about HCl, NaCl, which becomes HCl here basically, but again, exchange of the proton, I have a higher concentration of Na, but I have a lower concentration of H plus, which is highest at the top, and Cl negative is roughly equal throughout the region. So because you have these competing gradients, you have the slowdown of the velocity for the second case. Now there's a subtle effect of pH dependent zeta potential, which can change the charge on the particle, but that's not the major factor. The majority of the velocity increase comes because of these competing gradients and supporting gradients. So again, you can use this to create different kind of exclusion zone speeds. This happens, and then more importantly, you can also see different structures of instabilities. So these instabilities, for instance, in NaOH, we typically always see 5 fingers. Obviously, these experiments can be sensitive sometimes, so it's not always exactly 5, but typically this tends to have around 5 fingers. Whereas in NaCl, we get 3 fingers. And again, you can see we can get very good agreement with experiments from our multiphysics simulations. And I should add, there is basically no fitting parameters in the system. We use all the values measured in the lab and reported in the literature. So there is no fitting parameter. There are some differences in the sense we are not able to get exact time matching with the experiments, but so it's a more of a qualitative match dynamically speaking, but we are able to capture generally most of the features. Now, to be able to explain the wavelength of these fingers, you have to do linear stability analysis, which becomes a little bit complicated because of all the coupled physics. So that is something we were not able to make analytical progress on. But overall, we were very satisfied that our simulations are able to capture experiments more or less very well. And the second part I want to highlight here was that more on top of this, the key feature we want to highlight is diffusive phoresis is often thought of as a slow phenomena. That it is something that's not going to be very strong when it comes to kind of real life scenarios. And our main point is that you can actually kind of track these velocities by doing particle simulations and so on. And what we basically see is that you can actually get velocity 10 times and even sometimes even higher than that by using sort of these instabilities to create flows and so on. So again, diffusive phoresis can be combined with other effects and create sort of this instability phenomena. All right. With that in mind, now I want to quickly summarize what we have discussed till now. So diffusive phoresis is the movement of particles in response to a concentration gradient of a solute. We also discussed the features of banding, and then we use that banding phenomena to kind of trigger this Rayleigh-Taylor instability by using an ion exchange membrane. I want to go towards the last part of the talk, which is basically how this happens and how can you think about patterns formation on an animal. So this story really began when one of my students, Ben Alessio, he's a former undergrad in the lab who's now doing his PhD at Stanford, saw this fish in the Birch Aquarium in San Diego. And it's pretty impressive that he saw this fish because I've gone to the aquarium myself and it's not easy to find. So he saw this fish and it reminded him all these hexagons and stripes, reminded him of a code he was writing on his computer. And so he started learning about this field of Turing patterns. And so Turing patterns are essentially how we describe patterns on these animals. And they're essentially a reaction diffusion instability. So Turing's question was very simple. His question was, obviously these patterns have formed because of biology and genetics and so on, but what is the chemical basis of this pattern formation? So his question was, typically if a system is diffusion dominated, so when fish is growing or an animal is growing, I'm not creating a sort of bulk mixing. So this is a diffusion dominated system, and diffusion typically promotes homogeneity. So if I have multi-components, they should all become uniform. So how can I have a heterogeneous pattern arise out of a homogeneous condition? And so his argument is if the species have different diffusibilities, and more importantly, if they're reacting with each other, there is this delicate dance between diffusion and reactions, and that can trigger a reaction diffusion instability. So imagine a multi-component system with uniform species, you slight trigger it, and basically it yields a new pattern, and that's the Turing pattern. So mathematically, you have this unsteady term, this diffusion term, and then you have a reaction mechanism. The magic happens in this reaction mechanism term. This is something I'll not address in this talk, but I'm happy to talk more in Q&A if somebody's interested. All right. So what can this model do? So you can see on the top, I discussed this sort of hexagon patterns. You can very easily create them in Turing patterns. There's this radial eye pattern coming out of a puffer fish. Again, you can create using different reaction mechanisms and different other conditions. There's this beautiful study by Nakamatsu et al. Where they ablated a fish and then tracked these patterns over time and used initial conditions as an example, and you can again simulate these patterns. But what you want you to see on the right-hand side, on the simulation side, is all of these patterns are diffuse, which makes sense because it's a reaction diffusion instability. And diffusion, if you think about it in your mind, is always going to be blurry because that's how diffusion is. It promotes shallow gradients. So our question was, how can you have sharp gradients in a diffusive system? And now I hope you can remember back to the story, which is diffusive phoresis is a convenient mechanism which can create these banded structures. And so that's basically a simple idea. So this pattern is anything but diffuse. So how can you create this pattern in a diffusive system? So the idea is very simple. Is these morphogens, they actually do follow Turing's theory. And then when the cells assemble, they assemble diffusively. So small biomolecules, they follow Turing's theory. And on top of that, chromatophores, the biological colloids, they move via diffusive phoresis. So we didn't just come up with this idea by ourselves. There are other things that led us to believe that. So for instance, this beautiful article on Annual Review of Genetics, this talks about this pre-patterning and patterning has been observed in different species. So this pre-patterning is like the small biomolecules and patterning is like the chromatophore diffusive vertically moving. Then I'll show this data quickly, but this paper from Ram et al. Nature Physics, where they use diffusive phoresis to functionally propel unrelated biological materials, they report that cargo patterns become sharper with increasing cargo size. This is actually exactly the same phenomena that we see when we include diffusive phoresis in Turing patterns. Chromatophores have been shown to be diffusive phoretic. And then finally, the data you have seen today all the experiments have been in water, but you can actually, they have been seen in biofilms, hydrogels. So we do think diffusive phoresis is possible in a realistic system. So with that in mind, let me go and show you the simulations. So the top equation is the usual Turing patterns, and then on the bottom equation, we add this m grad c advection term, which is again a concentration of the solute driving the motion of the particle. Now I'll play some videos. Please note these are falsely colored, so they are not exactly one-to-one match. It's the same model, Turing model, but in one case we are simulating the species, in the other we are simulating the particle. This is just to show you visually what is the difference between when you do include diffusive phoresis. So you will see this video where you have with diffusive phoresis and without diffusive phoresis, and you will see all of these patterns become sharper when you do include diffusive phoresis. Now I want you to focus on the top left image. The top left image essentially is hexagons, and lambda c is the size of the hexagon, and is lambda n's, this video will come up again, is actually the lambda n is the size of the thickness of the hexagon. So thickness of the hexagon is lambda n. So there is a pattern size and then there is a pattern thickness. And so pattern size is determined as lambda c, pattern thickness is determined as lambda n. I'll come back to this parameter later when we discuss some results. So we call this phenomena diffusive phoresis and Turing patterns. Luckily, there were experimental data on the system. So these experiments were done in Ram et al., where they used E. coli to create these patterns. And then they propelled DNA origami, and then they added streptavidin to get a fluorescent signal. So they increased cargo number in their experiments, and they showed this sort of increased contrast between the foreground and the background. So when you increase cargo number, the particle, which is the DNA origami, the diffusivity goes down because bigger particles have lower diffusivities. So the Peckley number goes up, which is what we use in our simulation. And so as you can see, the contrast increases again, also in our simulations. Now, this is a qualitative comparison. There is no means to say there is any quantitative comparison, but again, it shows that including diffusive phoresis in Turing patterns is a convenient mechanism to get some of these features back. Next, we basically call this phenomena diffusive phoresis and enhanced Turing patterns. And then there's a recent paper which also even talks about diffusive phoresis within a cell. So we're talking about cellular assembly, but there is also some arguments about how reaction diffusion driven diffusive phoresis can create intracellular organization. If you're interested, you're welcome to check out this ACS Nano paper. All right. And finally, I wanted to also finally mention in the same vein, we wanted to now go towards particle modeling. So what you saw till now were all continuum results. So Siamak, who is an expert in particle modeling, kind of led this work. So now imagine you have background Turing patterns, and on top of that, you want to assemble the cells. So instead of representing them through continuum equations, now we are basically modeling each particle with a particle equation, which is given. So we have a diffusive phoretic term, we have Brownian motion, and the intermolecular particle interaction. In this case, these were just hard spheres. Now, in this case, we are doing one-way coupling. So the particles are responding to the Turing patterns, but they're not modifying the Turing patterns. I should mention there are ways to do that. For instance, this very nice paper from Zach Sherman's group at UW Seattle. However, these are very computationally intensive methods, and that's something our group does not have expertise in, but that's something we would like to go towards. These are just imagine particles responding to this pattern. So now this is how it works. So what you're seeing, chemical concentration of two species using different parameters in the Turing model. You get sort of hexagons and stripes, the usual things. Then we have two cells. They're assembling at different Peckley numbers, Peckley number of 1 and Peckley number of 20. And you can see we can create these different patterns. Here, what you see again, I want to point out now that the yellow cells might get trapped with the blue cells. And so these are some of the things that we start to see when you have finite particle sizes. Now, whenever you have finite particle sizes, you introduce another length scale in the system. So this length scale is particle size by the pattern size, so A by lambda C. So that's the one factor that comes, and the other is the volume fraction. So you start to get these two more parameters, and using that, we created a phase diagram. And so lambda n again is the pattern thickness, and lambda c is the pattern size. Initially, it was only a function of Peckley number in the continuum model, but now we can see it is also a function of volume fraction, which makes sense. So after one point, you cannot just pack any number of particles because they are finite size. So you start to sort of, kind of limit your thickness of these patterns. And more importantly, when you start to vary with this A by lambda C, you start to see this imperfection. So we have a similar pattern here. You see all three of them are similar figures, but we are varying this A by lambda C, And you can see, we can start to see this texture arising, this pattern breakup. And to the best of our knowledge, this is the first way you can get imperfect Turing patterns just by introducing diffusive phoresis of a finite particle size. This is something we're very excited by. In the future, we're interested in sort of thinking about cell-cell interaction in more complicated forms. We also want to include addition forces with the media, and also include things like phase separation, which are currently thought of as separate to Turing patterns, but we think should be combined in the similar framework. With that, I hope I've convinced you of three things in this talk about what is diffusive phoresis. They can trigger two types of instabilities, at least Rayleigh-Taylor instability and the reaction diffusion instability. And in the end, I'll just leave you with one last simulation of diffusive phoresis, and here you go. I'm happy to take any questions.